Yeah, I think we can start. At the same time, we will let people in. Good morning, everyone. Welcome to the 2021 Annual Translational and Transformative Informatics Symposium. I'm Zechen Chong, Assistant Professor of Genetics and Informatics Institute at UAB. It's my great honor to chair this year's virtual meeting. Thank you all for participating in this year's ATTIS. Some of you may recall, after last year's ATTIS, the COVID-19 pandemic began, and we started the long work from home working style. This is definitely a challenging year for everyone. Well, with more people vaccinated, hopefully after this meeting, I think that's what everyone's hope. We may start to return to work as usual. This year's ATTIS were sponsored by UAB Informatics Institute and the CCTS. First, I would like to thank the support from our leaders, Dr. Jim Seminole, Director of the Informatics Institute, Dr. Jake Chen, Associate Director of the Informatics Institute, and Dr. Bob Kimberly, Director of CCTS. Next, I would also like to thank all the program community members and the moderators. Specifically, I would like to thank the staff members for their professional, efficient, and tireless work. Thank you, Nafisa, Heather, Jennifer, Katie, and Amanda. Without your hard work, we will not have this meeting. Here is the overview of this year's agenda. My apology for the tight schedule. In a minute, we will have two great keynote talks. One is from Dr. Heng, the other is from Dr. Nick Navin. We will also have three one-hour talk sessions, two after the keynote talks, one in the afternoon. In each session, we will have 4 great talks from selected abstracts and the invited speakers. The talks include the biomedical methods and the biomedical applications. I guarantee you will learn a lot from these talks. At noon, we will have a quick lunch break, and you can grab your lunch to attend the biomedics education special session. We invited Philip Campillo from CMU, Chindo Hicks from LSU, Elliot Lefkowitz from UAB to initiate the discussion in this session. We hope you all can attend, and we would like to hear your biomedics needs and your opinions on how to better educate next generation biomedics researchers. In the afternoon, from 2:30 to 4:30, we will have 2 concurrent workshops. One is for single cell genomics. The other is about cloud computing for biomedical research. The 2 workshops will have hands-on practice. The cloud platform hosts large volumes of sequencing and clinical data and workflows to analyze these data. If you would like to give a try of biomedics, even without computational skills, these 2 workshops are for you. Please note the Zoom link for the cloud computing workshop is different from the main event. After workshops, our Associate Director Jake Chen and the Director Jim Seminole will give the closing remarks. I will post in the chat a web page, including the detailed program and the Zoom links. So here is the overall agenda. I hope you all can enjoy this year's ATTIS. Thank you. Is Dr. Heng here? I think Heng,
I'm here.
You can start to share your screen.
Okay.
Great. I can see your screen now.
Good. Thank you.
So first, I would like to introduce our first keynote speaker, Dr. Heng Li. Heng is a legend, I would say, in biomedics, a role model of mine. The software he developed, including BWA series, BWA, BWSW, BWMAM, some tools, MAQ, Minimap2, etc., are biometicians' everyday jobs. These tools are highly cited. For example, both some tools and the BW series has been cited over 31,000 times. They are the standard pipelines for many consortium projects, 1,000 genome, TCGA, ICGC, TopMed, you name it. Because of his excellent contribution to biomedics and science, he was awarded the prestigious Benjamin Franklin Award in 2012. Besides this, another legendary thing I would say is his fun of publishing single author papers. Given the current genomics collaborative flavor, I think this is pretty rare, if not impossible. Some of these highly cited tools he developed were actually his single author papers. Heng earned his bachelor degree in physics at Nanjing University in 2001. It's one of the top universities in China. He received his Ph.D. from the Chinese Academy of Sciences in 2006. Then he worked with Dr. Richard Durbin at Sanger Institute. In 2009, he joined Broad Institute and worked with Dr. Reck and Dr. David Altschuler. Heng joined Harvard Medical School as an assistant professor in 2018. He's currently working on biomedics algorithm development, molecular evolution, population genetics, and single-cell genomics. Today, he will talk about their most recent work on long-read pangenome de novo assembly. Let's welcome Heng.
Thank you. Thank you for the introduction, and it is my pleasure and also honor to present my recent work here. Today I'm going to talk about the title is the assembly of a human pangenome. It's actually two topics. The first the assembly, and second is the human pangenome. I think actually more than 10 years ago, I wrote published this review. It actually is on short-read mapping, but this is on the right. I'm showing the last paragraph. This is effectively is saying that currently at that time, 10 years ago, we were mostly working with a single genome, and so I said that I think we will be able to do alignments against multiple genomes. And then it basically has a pangenome. At that time, there's already work in that direction, but there's not much going on, in the past 10 years. And so it is, although the idea is new, but why that doesn't happening? And why, I think it's a time to really to think about pangenome now. I think one of the key reasons is that the high quality long-read assemblies. You can think 10 years ago, we can assemble genome with Sanger read, but still it's very expensive. There are only a couple of genomes at that time. There's only the human genome, human reference genome, and also the vector genome. There were just two assemblies. And although short reads, we can do lots of short reads human assemblies these days, but the quality is not as good, far lower than the Sanger read assembly. But now it's different. Now we have long reads. And so before I go into the talk, I will just give a very short primer on the different types of long read assembly. And so on in this diagram, and on the top, this shows a real diploid genome. This you have this the red and blue dots are basically two heterozygous allele. And so this the traditional assembler, most of the assembler, I would say 98% of the assembler will do this. When they generate a assembly, it will randomly mix the paternal and the maternal alleles. You see random switches on one contig. And this set of contig only represent one haplotype. It doesn't represent a whole diploid genome. And so a bit later, this Falcon and Zip published, that's 2016. And so people can start generating, we call this the primary assembly. And on the primary assembly, you see you see long blocks of long phase blocks with rare switches. You see from this red allele, a block of red allele switch into a block of blue allele. And so in Falcon and Zip, there's also a concept this called alternate assemblies. These are phase correct, but they are very short. They only cover the heterozygous region. It doesn't cover the whole homozygous region. And so the alternate assembly by itself is not very useful. It's very fragmented, and also it's incomplete. It doesn't have the homozygous region. And then 2 years later, Sergey from the NGRI now, published this called Trio Canoe. And the basic idea is it used the parental shell rays, used k-mers to pull apart the long rays, basically partition the long rays into two groups. One from the paternal haplotype and the other from the maternal haplotype, and assemble separately. And so they can do this, I call this haplotype resolved assembly. Basically, get very long phase blocks because you have the parental information. And so there are two problems. One, first is you need the parental information, and it's not always available. And secondly, at the point of the publication, they were using this noisy PacBio long rates. And so at that time, they can only get 1 megabase counting for the human genome. And it's very short. For Falcon and ZFIS, the primary assembly, we can typically get over 10 megabase, 10 to 20 megabase at that time. And so 1 megabase is much shorter than that. And so although we can get this assembly, but the assembly is fragmented is not as useful. And so what's the root problem to this? Why we can't get good assembly is that it mostly because of the high sequencing error rate. And for human, the heterozygosity, basically the chance to see heterozygosity in the human genome, that's about 0.1%. And the sequencing error rate at that time, at a couple of years ago, that was 5 to 10%. That's 50 times higher than the real human heterozygosity. And so due to this high sequencing error rate, there are two consequences. The first is it's difficult to get phasing right. It's even in the Trio Canoe paper, there are still, often facing errors because, the noise is very noisy. You can't have fully separated two haplotype. And the second problem is actually, it's a bigger problem that's also happened to, to collapse assembly, not just to phase assembly. In this diagram, I'm showing this a true haplotype. You think this is a haploid genome. This is not heterozygote anymore. It's a haploid genome. And so this blue and the purple part are unique in the genome. And then there's two red arrow. These blocks are tandem duplications. But because there's during the evolution after the separation of this, after the duplication, there can be small variance between the two copies. For example, this dark green block shows this is a difference. Think about it as a SNP between the two copies. And so you have sequencing rates, these long rates, and they will do the assembly. And most of the current assemblers will do this. It will either create a misassembly because it's because this the rates have high error rate. Remember this 5 to 10% error rate, and there's just a single SNP separating the two haplotype. And so it's very difficult to really see the SNP. And so people, the assembler will suggest saying, oh, they are actually the same thing. And so they will either produce a misassembly that collapse the two copies together. Or, there's some smarter assembler will see the assembly graph is sort of weird, basically because of this rate. And so it will see, oh, the assembly graph is unusual. And so there might be something happening. And so they will break the assembly. Then they will introduce a assembly gap. And so, but in either case, you will lose one copy of the gene. The most of the assembler will do this. In these tandem duplications. And so the things have changed in the past, just past 1 year, I would say. The PacBio now have this called high-fi rate, high fidelity rates. They are long and accurate, and they are in telomere to telomere assembly. And so here I'm showing this the rate length and also the error rate. You can see the universal shell rate that was came to the market in 2008. It's about a couple hundred base pair. These days, I think the dominant length is 150 base pair, and the error rate is below 1% in the average. And then 5 years later, people have long rates noisy, and these days you can even get 1 megabase long rates from the nanopore. They are very long, but you can see the error rate is very high. This is about 5 to 10% error rate. And then the high-fi rates, although it's not as long as noisy rates, it's much longer than the shell rate. It's around 20 kb these days. And the error rate, actually, I underestimate the error rate. The error rate should be even lower. These days, it can even reach 0.1%. And so I call this a breakthrough every 5 years. it's I mean from 2008, 2013, and 2019, and most existing assemblers, long range assemblers are divided for this type of rates, and they are not taking advantage of these rates. And so if you use these existing assemblers on these rates, you won't get much better assembly, but you won't really see the breakthrough. And so that's why we developed this HiFi-SM, this new assembler. It's now published in Nature Method in February. I think this the HiFi-SM, and the key feature of HiFi-SM is take advantage of the high accuracy, and so it can resolve the previous example, the sectional duplications, and lead to better assemblies. And here on the right, it basically shows a workflow. It's very similar to the current the conventional assemblers. It's first do oversaw read overlapping, find the overlap between all rates, and it's that face aware error correction. That's this part is different from the traditional assembler. Next slide I will explain what face aware error correction, and then that's the layout and then to generate the content. And so this is why I think this habitat aware part, the habitat aware error correction, is one of the key things that separates HiFi-SM from the rest, the most other long rate assemblers. And so basic assumption is there are very few recurrent errors in the sequencing in the long rates, and that is can be applied to HiFi rates. The sequencing error rate is very low, and it's you rarely see recurrent errors. And the procedure is something like this. For each rate, you choose a target rate, and then you find all the overlapping rates. This red one is target rates, and this red and blue ones are other rates. You find all the overlap between all rates, and then you identify this. I call this informed sites. These sites are supported by at least 3 other rates. You can see the red one is supported by 4 rates in total, and so there's 3 rates, and so this blue allele are supported by 3 other rates. These are called informed sites. And in contrast, this is not informed sites, because the green allele is supported by this 5 rates, but the pink allele, this most likely sequencing errors only supported by 2 rates. And so these are not informed. Only this one is the informed sites. And when we can identify the rates. The rate that's on the same phase as the target rates. In this case, the red rates, because they have we identify these rates by only looking at the informed sites. We don't look at these sites. If you look at this side, oh, these 3 rates are not on the same habitat. But if you look at the informed sites, you have enough information, then you can see, oh, the target rate, the black one, is actually belong to the red habitat. And when you have this, you can do the error correction only using the red rates. You ignore the blue rates during the error correction. And then you will identify, oh, this is sequencing error. You can use this correct allele to fix this sequencing error. This is this way, we won't basically correct true heterozygotes away. We will correct sequencing errors away, but we will return most of the heterozygotes. Also, the overlapping phase is different. I will skip this briefly. It's similar to the previous overlap. This OLC, overlap consensus layout consensus algorithm, but it has a habitat information, and it's with aim to build two habitat. After assembly, you will get separate habitat for each habitat. It's not like the traditional one. You will mix the two together. And so also with the here I'm showing this real assembly graph. This after this layout procedure, and here I'm still using the parental information. Although these days, right now, and HiFi has also you can use Hi-C to do the similar things. But in this example, I'm still using the parental information. And so you have this the red allele. Each bar is a long bar is we call the unity. You can think as a shorter context, and the red bar coming from the maternal habitat, and the blue bar come from the paternal habitat. And the gray bar is the homozygous regions. We basically, with the parental information, we can identify all this. And the next step, to spell the final content is quite easy. You just traverse these red and gray bars. You traverse it, you will get a 2 megabase long content for the maternal for the maternal haplotype, and it's complete in this example. And also, if you trace the blue part, then you will get a 2 megabase counting for the paternal assembly. This is basically how hepatome works. And so this on the real data, this on showing this bunch of other species. This is human, and you can see the hepatome is consistently produce higher, longer contigs than all the other assemblers. There's hepatome. Hekanu is the only other, so far is only other assembler that takes advantage of the high-fi accuracy information. And the peregrine and falcon, although they can do this type of assembly, you can see the N50 is much shorter than hepatome. And also, I talked about in previous slide. I talked about the problem that you may have misassemblies around sectional duplications like this. Assemblers often have mistakes around these regions, and so we came up with a measurement to measure how often these type of things happen. Basically, we call it the percent MMC, is percent multi-copy genes in the reference that are missing in the assembly. And suppose there's a genes in these sectional duplications, and because it's dupe, and so there will be 2 copies in this haplotype. And if you either have a misassembly, you collapse the 2 copies, or you have an assembly gap, you will miss this gene. And so this lower percent, if you miss fewer genes, then it's a good assembly. And so with this matrix, and you can see this is the assembly of the CM13, this high-polygy genome. And so we know the ground truth. The T2T telomere to telomere consortium has a ground truth for this assembly. And you can see this is the assembly. This is on the y-axis. This is the percent MMC, the percent missing copy genes. The lower is better. If it's 0, means there's no error. And if it's 100%, it means that it misassembled all the segmental duplications. And you can see here the different assembler, different data type. This Oxford nanopore, and this is the packball continuous long reads. This older packball data. This is the high-fi reads. You can see that hepatome almost make no error. Actually, there's only 2 missing genes, only miss 2 multi-copy genes in the entire human assembly. And the Hekanu is almost as good. You can see the other assemblers, 20%, 70%. If you use other assemblers, they will miss multiple copy genes. And just imagine if you use these type of assemblies, you won't be able to study these multi-gene families. Most of these genes have multiple copies and in several duplications. You can only do that with hepatome and or Hekanu. And the previous example, this shows this CM13 basically assemble the same. We have the ground truth, and in this case, we don't have the ground truth. This is a different sample, 800733. This is a different sample from the ground truth. You can see, I mean, this even use the GRC38, you will see around 10% MMC, meaning that 10% of the GRC38 multi-copy genes and 10% of the multi-copy genes in CM13 is missing in GRC38. And if we trust GRC38 is a good assembly, then the 10% is just a background. These are the true rate of multi-copy genes in the GRC38. And you can see that in here, the hepatome and Hekanu, they're also about 10%. Also, this is a different sample, which means that they are still pretty good. And if you look at others, I mean, this story is similar. They have much higher rate of misassemblies in some duplications. This is showing that the hepatome is and also the Hekanu is produced much better assembly than all the previous ones. And so hepatome, one or two best hepatome assemblers, the other one is Hekanu, and vastly outperforming all the other long-term rate assemblers. And it's also so far is better than Hekanu on hepatome resolve assembly. And so far, hepatome is the assembler of choice by the Human Pangenome Project. I will talk a little bit later. This the Human Pangenome Project is using hepatome as the only assembler to produce the assemblies for the year-round data. And this is based on the evaluation of 23 with the 23 other assemblers and pipelines, and hepatitis clearly stands out. And this unanimous people, I mean, the consortium unanimous think that hepatitis is the best assembler for this job. And in addition, hepatitis can do metagenome assembly. This is unpublished. Okay, they move to the second half. The pangenome graphs. I mean, for pangenome graph, I will start with actually a problem with the current. I mean, the current model for the human to describe the human genetic variations. And so I will start with this and how to encode the human genomic variations. And so the existing solutions you recall is as the substitution model. I mean, the substitution model here, I mean, both HGVS and also the VCF format are like this. And for those who are not familiar, this is the VCF format here. I'm showing it here. And you have the chromosome and position, and this is the reference allele is substituted by a alternate allele. In this case, the reference G, and you substitute G with this A. Basically, you specify the coordinate and substitute the reference allele. I call it the substitution model. It's great for SNPs. You can see it very clear what this SNP is. And it mostly works for indels, and here you can see the indels, and it works has some minor issues. I mean, it's mostly because human diploid, and it's there's phasing issues, but I mean not big issues, but it's inadequate for complex events. And so I'm showing here. I mean, we use the example multiple times, and so I will just introduce this locus. This is the human complement component 4 region. This is, you can see on the top. This is from secretary from 5 years ago. This is the human HRE region. The whole region is about 5 megabase, and there's a class 1 HR genes. This class 1 the serial A, B, C, the class 1, and so class 2, the DQ gene and DR genes, class 2. And C4 is sitting right between these two clusters. And this is called the class 3 genes. This is C4A, C4B, the homologous genes. They are very similar to each other. And so here this shows the C4, both of them have the similar structure. C4A or C4B, they have a similar structure. And they have 41 exons. This is very long. I mean, at least in terms of exons, they have many exons. And there are also differences. And also in these genes, there's a structural variations in this gene that between exon 9 and exon 10. This HRV, this is a virus-like transposon. And some of humans have this HERV insertions, and some others they don't. And so the C4A and C4B differ mostly defined by 4 amino acid chain changes. You can see the 4 SNPs. This the 1, 2, 3, 4, the 4 SNP chain changes, and they lead to 4 amino acid chain changes. C4A and C4B are the difference mostly defined by this. And so in the human population, as here this shows the in that paper, it shows the count of the different haplotypes. And this is the reference haplotype. This AL means long form, and the long form means it has this HERV insertion. You can see this dark greenish things here. This means that it has this HERV insertion, and if it doesn't, then it's called short form. And so the human reference genome, this is human reference genome, and this is the dominant haplotype, and it's AL C4A long form, BL C4B long form, and so it has two copies, and both of them are long form, and this is most of the dominant one according to the paper. And then there's some other this I believe this is actually a mislabeling in the paper. This is really AL and AS if you trust the color, because this I mean you can see this A short form AAS, AL and AS, and here the AL, and someone only have B, and then you can see this rare haplotypes, and someone may have 3 copies and 2 copies of this, and someone may have 3 copies of A but doesn't have B. There's a huge variation around this locus. And then I'm thinking about the VCF. I mean, how you would be able to encode this type of variation in VCF? These are changing in copies, and actually each copies are different. It's impossible to encode this type of information in the VCF. And previously it's okay, because I mean we were not able to assemble through these regions. Only I mean we only see it in reference and a couple of very good assemblies. Most have been misses. It's okay. But now we can do the assembly. This type of system we can very confidently assemble through these regions. Now how do we represent this type of variations? We can't use this anymore. And so this is then we came up this a graph. A graphical representation. And so I will go through this example. And here it shows each long block represents a long sequence, and the red one coming from the GRC38. This is the red one. If you trace the path, you trace the arrow, the dotted arrow here, you will get the reference, the GRC38 habitat. And remember that GRC38 has a two long forms, and so this 600 base pair thing, this HERV insertion, this is another HERV. I mean, this is all HERV. You can see the length is the same. They're very close. I mean, they're very similar to each other. And then if you trace the dotted line, you can see this is C4A. This is A genes with long form because it has this HERV. And then this is C4B, also a long form. It has the HERV. It goes through here. Then you see this is the reference habitat, and you can see then you can find other habitats. And if you go through here, instead of I mean this large part, you just trace here, go through here, and then here you basically cut out the whole this part. Then it will be only either a short form B. I mean, you will only have one copy. And depending on whether you have this HERV insertion, you will have long form or short form. And in some I mean more extreme case, if you go through this red allele, this actually you will have 3 copies of C4 genes. And then it also depends on whether you have this insertion, you can have long form, you can also have short form. This graph encodes most of, not 100% information, but you encode most of the information in this graph. And so this is way we think this should be a way going forward to describe this complex loci. And this is generated automatically. And by the way, I mean, why the C4 is important, and this paper is published in Nature Genetics, why it's important. I think so far is the strongest shows the strongest association with schizophrenia. This is also showed with a few other phenotypes. So the solution, basically, so I describe this graph, and then how the next question is how to generate it. And the basic idea is actually quite simple. We just take, I mean, if we have a collection of say C4 sequence from the C4 regions, we just collapse similar regions onto a node in the sequence graph, and in this case, if this part means that all sequences are similar around here, we just collapse it, and then I mean you will get this graph. But the devil is in the details. It's not that trivial to, I mean, how excited to do that is not trivial. That's why we have this, basically in the past decade, we have been dealing with this issue. And I also need to mention here, a side note is that this is not the only solutions. And you can build an FIB index to index all the sequences. There are pros and cons with that type of methods. And the advantage of full text indexing is that it keeps all the information. And the cons with the full text indexing is that you don't have a structure. You don't have this type of visualization. And so it's mostly for storage and can be used for alignments, but it's harder to interpret it from a biological perspective. This is how my program, I published mini graph, I think more than a year, less than a year ago in genome biology. And this what the approach that we are taking to generate a pangenome graph. And the idea is actually also very simple. And it's collapsed the orthologous sequences and only considered structural variations, the events longer than 50 base pair. And on the right, I'm showing a diagram to show how this really works. And basically, you start with a reference genome, this assembly 1. You think this as a reference genome, this is GRC38. This is the first assembly. And mine only have one assembly. Then this is then the graph only has one assembly. Then you have you have another sample. You have this ASM2, this blue contig. You align the contig to the graph, and in BLD in this case, the graph only has one JR38, and so this is just pairwise sequence alignment, very standard pairwise sequence alignment. You align it, and then you will identify the regions that are very different between the blue assembly and the red assembly. In this case, you have this we call the bubble, and you see the bubble. You max out the regions that align well. You cut. I mean, you only return the very, the divergent region, and you insert this part into the existing graph, and then the new graph encoding two assembly will look like this. You still have the reference path, and then you have these parts representing this structural variation. And here it's mostly a insertion. Then after the second assembly, you get this graph. And when you have third assembly, you do the same. You align the sequence to the graph, and this time this is not pairwise alignment. Because you have a graph, you need to do sequence to graph alignment. And when you do the alignment, similarly, you will identify, oh, there are some regions that's very different from either assembly 1 and assembly 2. And so again, you insert this part to the assembly, then you have a graph encoding 3 assemblies, and you just do this iteratively. For each assembly, then you will, in the end, you will get a assembly graph encoding at least the structural variation, these larger events across all the input assembly. And the thing about it is, this is actually very similar to this partial order alignment. This POA means the partial order alignment. It's widely used in sequence, multiple sequence alignment, and also used in error correction. Actually, if you look at this, the rationale behind this is the same. The detail is different because the POA is doing the base alignment, but here we are not really doing the base alignment. We are only returning the structural variations. And this is basically how minigraph works to generate a pangenome graph. And so we actually missing slides. Then it's we applied this approach to the data produced by the human pangenome graph. And so far the human pangenome project. The human pangenome project, so far they have these days the latest number is 90 haplotypes. There's 45 human samples. Because we are only doing the phase assembly, and so 45 samples, 90 haplotypes. And at the time of, for this slide, I'm using a slightly older version, and there are 42 samples or 84 haplotypes here. And so here we use the JRC38 as the base, as the first assembly or the reference assembly. Then the next assembly is semester 13. This is the assembly produced by the telomere to telomere consortium. It's nearly complete assembly. And so we have 41 other deployed samples from the human pangenome reference consortium, HKRC, or human pangenome project. And these data are all assembled by TRIO FSM using the parental information. And this is the best deployed. Each one of them is the best deployed human assembly so far. This so far at the gene bank is far better than any assembly in the any whole genome shotgun WGS whole genome shotgun assemblies in gene bank, except of course JR38 is better, but it's better than all the other human assemblies. And it takes about 40 work hours to generate this graph, so it's reasonably efficient, not that fast, but it's reasonably efficient. And so this graph shows, remember that this is an iterative approach. And so each step, you will insert some new structural variations into the graph. And so this graph shows how the count of the insertion after each sample is added. On the y-axis, this is the number of events. You can see this number of structural variations, different types of structural variations inserted to the graph. On the x-axis, this shows the sample. This is the first sample, the second sample, the third sample. And the first bar, is showing that, remember this, we start with JARG38. The first sample to insert is this segment 13. And this first bar shows that after you insert the segment 13, you will see that you inserted, this is 12,000 events into the graph. And then when you insert the second habitat, because a lot of common structural variations are already returned in the segment 13, and so you won't insert again. And so after you insert the second sample, you only see 6,000. This is half. This is from 12,000 to 6,000, which means a lot of structural variations is actually already present just a couple of samples, common structural variations. And so this when you insert second sample, you will insert 6,000 events, and third sample, after you insert the third sample, you will insert 4,000 new events on top of the previous graph, and then you just do this, and so you can see that there's some spikes because it's actually changing the population. And the Puerto Ricans, this admin sample, so they have some different components. Also Africans, they have much higher diversity. And so when you start to insert Africans, you will immediately see a lot of new structural variation not seen in the rest of the world, only present in Africa. That's why you see these spikes. And for these mostly non-African samples, you can see this only several hundred events, which means most of the common structural variants are already encoded in the graph. Then here you don't only insert small events. And even for Africa, you can see the trends gradually is going down. With each new sample, you will see fewer new events. And after this procedure, this what a real graph look like. This is the 500 kilobase region coming from the segment 30 from the chromosome 11 around 20 base region. The whole graph will typically look like this. This look and feel look like this. This again the red bars. This is the JRG38. And because each these small ones encode a structural variation. And remember, we use the 50 base pair as the threshold. And in the following, I will call it bubbles. This is the common terminology in assembly and also in sequence graph. This type of thing we call it bubble. Basically, it's in the simple case, it's simple bubble. There's only two paths. And there's some complex bubble, you may see more than two pass through it. But overall, this look like this. And you can usually interpret one bubble as one variation. And so this is, multi-allelic variation. This is a biallelic variation. And here is biallelic. This is an insertion here, 80 base pair insertion here. And so overall, this graph is it has 80,000 bubbles of variations. It involves around 75 megabase of JRG38 in the bubble. You will think it's a little bit more than 2%. And this is coming from 84 habitats, and they are actually only 2% of the JRG38 are influenced by the bubbles. And most of them, in terms of count, most of them are biallelic. But some are triallelic or tetrallelic or higher. The tetra is 4 alleles or higher. But if you look at it, I will come back to this a bit later. You can see these alleles actually cover, in terms of length, it covers a lot more length than the biallelic alleles. Biallelic, there are a lot of them, but they are very short. And these ones, although they are fewer by counts, they are much longer. And so it's this basically shows the classification, the repeated content of these bubbles. Basically, you extract the bubbles and identify this the LU, L1, this is the repeated class, bin here, and as a satellite, you can annotate them. And you can see 20 by count, 25% are LUs. This is a 300 base pair interspersed repeats. They're moving around the most and most of them are these, and most of them are biallelic. The red bar show this is a biallelic allele, and this is the blue means triadic, 3 alleles. This is 4 alleles or more. This show the green, and most of them are biallelic. And also the L1. This is 2 most active class of interspersed repeat in the human. And so there are others. And you can see what's striking is the bin here. VNTR, this variable number of repeat by definition is over 6 bit. The repeat unit is longer than 6 bit pair, which in poly A is not VNTR. Because in poly A, the length of the motif is only 1 bit pair, and here we require 6 bit pair, 7 bit pair or longer for this one. And you can see this in terms of the count, most of them are actually dominated by biatic allele, but this part is different. And what's happening here, I'm going to show you example why this part is different. This is also a region on the human chromosome 1. And you can see in this region, the different haplotypes. You can see this different samples. And you can see this, if you are familiar with IGV, this small number, the 504, is showing the length of the insertion. And for system 1, you can see this 5 base pair insertion, 100 base pair insertion, 400 base pair insertion, 700 base pair insertion. But you can see that they're inserted all around. It's not like, I mean, some at the same local, they're all around. And you can see the length all different. And this is not just human in this say orangutan chimpanzee. Although these are not insertion, but they have deletion on also of the different lengths. And this shows the graph is a mass. It's basically you have, because there's all the, I mean, most of the human actually have it's special specific allele, the private allele. And so this graph is massive only, and you shouldn't look too much into this graph. It's just showing the complexity. And so you can see in this part, there will be multiple passes through the graph, and that's why it has multiple alleles. That explains this part. I mean, most of the VNTR will look like not as complex as this, but I mean, they have multiple alleles. And this figure shows the distribution along the chromosomes of different events. There's the red shows the interspersed repeats like LU, L1. They are roughly evenly distributed along the genome. And then the blue, this is the VNTR. You can see this all center, I mean, all highly enriched towards the end of the telomeres. And then this green are also similar. I mean, this is a very special class of repeat VNTRs. And so the above shows the by count, and if you by length, it's actually a very different plot here. And here is I mean, how this plot is generated is basically if there's a event, you calculate the sum of length of the reference allele in this bubble. And so in terms of length, you can see that LU is much shorter because LU is very short, only 300 base pair. And so if you sum over all the G38 sequences involved in alleles, I mean, although there's a lot of them, but because LU are short, so you actually have lower bar. And although there are much fewer L1s, L1 is the longest L1 is 6,000 base pair. Although most of the insertions are shorter, but still, I mean, if you sum of them, then I mean, there are more L1s in G38 than LUs in terms of the length, and similar with the others. But what's striking is actually these bars. I mean, these bars, the satellites, mostly the centromeres, I mean, they're pretty small by count. You can see there's a lot more of them. These are the centromere sequences. And then these VNTRs, also, I mean, although there's a lot of by counts, and that also you can see this by length is also longer. And although the VNTRs is you can see by counts, I mean, the length of the height of the red part is actually quite high, but here is almost invisible, which means most of the VNTRs are very long and very complex. And also this bar, this partial repeats, this mostly involve the segmental duplications. This is even more striking. Because here, I mean, the red bar coverage is quite high. The green part is very small, but here is by length. Most of these lines coming from these complex regions. And so, what are these regions? And in the rest of the talk, I will go through several examples. I mean, what's happening in this region. Most of, I mean, all my examples in the following will come from these regions. And so the first example is a relatively simple one. This is TNNT3. This is, I think, this important gene that is developmentally in important genes. And this gene is actually what we believe is that it's misassembled in GR38. Now we have a better assembly. In some areas, we have better assembly than GR38. And so on the GR38, the scheme is like this. There's a TNNT3. There are 20 kb repeats at the upstream of the TNNT3. But in semester 13, it's reversed. The 20 kb repeat is on the 3' end, on the downstream of TNT3. And if I think if you study this gene, you will think, oh, this is the promoter regions, but it's actually wrong. These two repeats are not the promoter region. This part are the promoter region. Basically, this part are the promoter regions. But with the assembly, we can identify this type of errors in GR38. If you study this gene, then just ignore here. The real deal is here. And this shows the RHD gene, and this is important gene that determines the RH group, blood group, and RH group blood group. You may hear sometimes you may see this is not only ABO this type. There's an A minus, and this minus is determined by this RH factor. This is mostly determined by this RHD genes. Basically, if an individual has functional RHD genes, then it will be plus type. With A plus means that they have functional RHD gene. If this doesn't have functional RHD gene, it will be A minus blood group. And so, and RHD is subject to frequent structural variations. And here shows the graph around the RHD gene. The graph is a bit complicated. It goes around here. You trace here, and then you go down here, loop it again. You leave this. This is the red part is the reference, and so this shows the reference haplotype. This has RSD. That's RC is a close homology of RSD gene, and so there's another gene in between. This gene RSD is here, and the RC. That is a dominant remnant haplotype. The remnant haplotype has the RSD gene, and then there's RS minus group. It doesn't have RSD gene at all. It only has RSD genes. It's actually quite frequent. In this sample, and then this I believe this is kind of a rare, it's very rare haplotype. I haven't really dig into the literature too much, but I believe this hasn't been documented. This is a duplication of the RTD gene. You have an actual RTD gene here. This is a completely copy. And it's, I don't know if it has promoter, but it has all the splicings. It's nearly identical to the relevant RTD genes. And then there's also a inversion. You have an inversion that involving both RTD and RTD genes. They actually flipping the content between the two genes. And then this I will quicker. It's the CYP2D6. This is a very important gene in drug metabolism. It has multiple copies, and the graph it looks like this. And again, the red one is the reference allele, and this is the CYP2D6. And then there's CYP2D7. This is pseudogene of CYP2D6. I think people believe it's non-functional pseudogenes. And so there's then you have multiple copies, and from the graph you can infer the frequency of the different. What's the frequency of the copy number changes, and this is from the literature. You can see this is about the same. It's a vary between Caucasian and African Americans, but you can see this we have more Africans, and so this is the frequency is actually closer to the African Americans. And similarly, this a gene associated with Alzheimer's. This you can see this again this copy number changes, and this is similar. You can infer the frequency from the graph, and the frequency is similar to the previous. They are doing the protein mass. It's not. They are not doing the DNA sequencing because it's very difficult to sequence this one. And in this case, the reference haplotype has two alleles, but the dominant haplotype only have one allele. This the GR3A is actually a very rare haplotype. And you can count this, and also here shows again the previous graph, and you can similar to the count, but it's actually slightly different from the previous one. Is that the count is actually different. And so we trust our results, but this could also be caused by population difference, because we tend to have more African samples here than their studies. This HRA gene, and much more complicated. And I want to highlight this gene, this HRAY gene. This is a pseudogene of HRA gene, and this HRAY gene is a quite young gene, new genes, not young gene, new genes. They found that 20 years ago, and is also observed in HGDP. At that time, I mean, we don't know where this gene is located. We know that it's very similar to HRA, but it's not HRA because it's a pseudogene. It doesn't have introns. But it's actually, once we have the assembly, it's very clear it's here. And this is actually quite common. You can see it is present in 27% of HRPRC sample. It's common habitat. And also this strategy is a unique region, quite a unique region in HRA. And previously, I mean, we just don't see this part, and now it's actually trivial to identify this. And RPA, this is another medically important genes, even more complex. I caution here, warning, this complexity should be real. It should be a very complex gene, but don't read too much into the topology. When this becomes this complex, I mean, the incremental procedure may not work very well. And it's also harder to assemble. I mean, for 4 samples, we failed to assemble this gene. But if you look at the previous assembly, as least I tried the gene bank, none of the previous assembly except the GR38 assembled this region. No one assembled this region before. And the GR38, and people usually believe this is a misassembly. It's much shorter than what it should be. And so these other protein genes, about 800 genes with coding regions overlapping with the variations, and 300 of them has complex structure. This includes some important genes, CYP2A, 2A6, CT48. These are all actually known cluster. People know this is complex, but previous people can't really go in too much into that because they don't have the sequence. And some regions are difficult to cluster, any cluster, and so. This data is available now. It is unrestricted public data. The data has been released, and the assembly is being uploaded to the gene bank. And so, in the next few months, probably you will see that from the gene bank, you can study this. And the graph is available here. And the consortium is building an even better graph with higher resolution, with base resolution. And so I decided the summary slides. I talk many things, and I think the 2 key messages I want to say is this. The first key message is that we have much better assembly than ever. It's much better than all the previous assembly. I mean, you can see that this is all the previous assemblies, and here is they all have this around 50% error rate, and these days near to 0 error rate around the segment multiplications. It's very important if you want to study the complex gene families, segment multiplications. This assembly will be the key. And secondly, the second message is that although you can, we can do the same calling with elimination rate, we can probably call most of the alleles and some of the small structural variations, but we can't do the structural variation for VNTRs. The VNTRs was found by the high variability of VNTR was found by Biomedical 2 years ago. That was using the long rates. In fact, it's not possible. I mean, it's always very limited to study the VNTRs with structural rates. You can do long rates, but even with long rates, you can't eradicate this bar. This is most of these are some duplications, and even long rate. If you just look at the long rates, you don't do assembly, you can't study this part. And with the better assembly so far, we can really study this. And I call the elephant in the room. It's in terms of length, most of the structural variations actually are enriched in this part. But because the previous is just too difficult to study, it's too difficult to assemble. People often just intentionally or unintentionally ignore them. But this, with this assembly, we can really study the gene. And you can see, I showed the example multiple these medically important genes actually are just fall in this bar, and now we can study them. Yep, I will. There's also challenges. I mean, we need to do better assemblies and also gene annotation. This all there's still a question. I mean, this very preliminary results. It shows it has potentials. But how to make it acceptable to biologists is still a question. We are working on that consortium with my colleagues. are still working on that. And at last, I want to thank people from my group. This Hao Yu and Chong, they doing the assembly. Collaborators Xiaopa now in University of Copenhagen, and Pegabel and Howen are also on the assembly. And then on the human pangenome project, Benedict from UCSD, Ira from Yale, they are the leaders of these projects, and also the working groups. They have, given me many useful feedbacks. And thank you. I will stop here.
Okay, thank you, Heng, for the great talk. Any questions? You can unmute yourself and ask right now. Yeah, excellent work. I have a question regarding the SV pangenome graph. Did you try to evaluate the sensitivity and the precision for the SV? You know, we have a consortium. I'm a member of that consortium. We are actually working on the SV detection. So do you have a sense of the sensitivity and precision through the pangenome graph approach?
Ira from Ira Hall, I mean, his group is evaluating that. I mean, so far the approach is a bit, it's a bit simplistic. It's using, it takes the deep call SV calling as the ground truth and to evaluate this, to evaluate the pangenome work. And I think for at least for simple tractable variations, I mean, it's very high because the methods are very similar. It's the mini graph is using Minimap2 code to do the alignment. And so as long as Minimap2 can see it, I mean, it's also sees it. And so this is, I say, the simplicity, that's the intercorrelation. And so in the next step, we will probably compare it to the HG-SVC result. We have overlapping samples, and we plan to evaluate that in comparison to HG-SVC because. But I believe I have briefly done that in the minigraph paper, and I think concordance is quite high. And most of the difference actually coming from the VNTRs because the different allele representation and also where to put the VNTRs, the coordinates of the VNTR can also vary. Most of the difference coming from those.
Yeah, you mentioned the VNTR region that show the example of complex insertions and could that be an alignment issue? You know, if there's a messy or there's a bunch of repetitive sequences, the alignment could be challenging, right? It's
both. Actually, I believe assembly should be correct in those regions, and so you see that mostly because the alignments. That's, I mean, it's difficult because these are not VNTRs. I mean, not very few VNTRs are exact. Repeat. I mean, the same motif. I mean, repeated multiple times. Most of these long insertions. I mean, if you look at the sequence, they are sort of similar, but they are not identical. And so then, I mean, it depends on the alignment parameter where to really put the insertion. Do you align it as one insertion or you align it as two insertions? And this all become problem. And so there's a lot of uncertainty there. I think that's just innate uncertainty and can't really be resolved.
Yeah, but actually the graph was based on the partial order alignments, right? That's also multiple sequence alignment. Is that better than the pairwise alignment? Oh, I think it's better than pairwise alignment, but
it's a very good question. I mean, they didn't say that. You can see, I mean, for pairwise alignment, you basically will put it everywhere, kind of random due to the uncertainty. And with the graph, it's actually a force all the things. You can see this is a long region, but the reference part involved, you can sum this only a bit more than 100 base pair, which means I mean, all these trial variations actually are they are forced into this 100 beta region. Basically, they will relate the listening together. I think this is the advantage of the graph approach.
Yeah, that's awesome. Okay, the final question. I think the Minimap tool stand alone as a standalone tool that target high fire rates. You know, the HDSVC use multiple platform, and there's a strategic and other types of sequencing data, and they use a kind of complex phasing approach. Can you comment a little bit? Is that better a strategy compared to mini assembler, or have you compared the results?
We haven't done. I mean, careful comparison to HDSVC. But I think this even for the assembly alignment, I mean, this is have this Unimap. This is a kind of a extension to Minimap. Not the extension, it's optimized for assembly alignments. Actually, Minimap 2 currently are missing quite a lot of long structural variations due to the algorithm. And for example, HDSVC use LRRA, it's better than Minimap 2 over this long. I mean, long enough. Although I mean, Unimap is comparable to that. But I mean, this all a bit preliminary results, and we have just generated the full graph, and we will compare to the HDSVC. Then, and at that time we will get more. I mean, from conclusion.
Okay, great. Thank you, Heng. Thank you very much. Great talk.
Thank you.
Yeah, Nick, probably you can share your screen now. So our next keynote speaker is Dr. Nick Navin from MD Anderson Cancer Center. Nick is also a legend, I would say. When he was a graduate student and worked with Dr. Michael Wigler at Cold Spring Harbor Laboratory, he developed the first single-cell DNA sequencing method. This pioneering work established the field of single-cell genomics. Inspired by this work, other single-cell DNA and RNA sequencing methods were developed subsequently. In 2011, he was directly recruited by MD Anderson Cancer Center as an assistant professor. Nick holds a dual appointment in the Department of Genetics and the Department of Biomedical and Computational Biology at MD Anderson. His work is focused on single-cell sequencing, cancer genomics, bioinformatics, and genome evolution in cancer. His group has recently published the sentinel work on cancer cell evolution using single-cell sequencing approaches. These were published in several high-profile journals, including Cell. There's multiple Cell papers published in his group, Cancer Cell, Cancer Research, Nature Communications, Genome Research, Genome Biology, etc. And just a couple of weeks ago, Nature paper. Congratulations, Nick. He received over 20 honors and awards from different cancer research foundations and institutes. Because of these outstanding contributions, he was early promoted as an associate professor in 2016. Today, the title of Nick's talk is Every Cell Has a Story to Tell. So I will stop here and let's welcome Nick to tell us about the story.
All right, thank you so much, Zechen, and thank you everyone for inviting me here to speak today at this symposium. Let me just go ahead and share my screen here first. Does everything look okay? Okay, great.
Yes.
So I would like to tell you a little bit today about our efforts to develop single-cell sequencing technologies, computational approaches, and their application to understand cancer evolution, especially as it pertains to breast cancer. And so the title of my talk is Every Cell Has a Story to Tell. And I think it's a fitting title. What it is intended to emphasize is that every cell is unique and diverse, and that by comparing cells, we can learn a lot about the history of cancer and the evolution of cancer. So I'm going to give a little bit of an overview on the background of the field of single-cell genomics and how much it's progressed over the last 10 years. I'll be talking about a recent paper that was published just recently in Nature on triple-negative breast cancer and primary tumor growth. And then thirdly, I'll talk about a computational method that we recently published called Copycat to infer copy number data from single-cell RNA data. So for a long time, we've been interested in how a single somatic cell in the tissue, in breast cancer, this would be an epithelial cell in the duct, can acquire mutations and then diverge genetically to form a tumor mass. And a big question that we're trying to address in my group is whether this genetic and genomic diversity that's generated along this lineage is just stochastic noise or due to genomic instability, which we do not think is the case, Or is it likely to be a substrate for evolution and for selection that could be important for biological processes in cancer, such as invasion, metastasis, and therapeutic resistance? So there have been different models proposed for how tumors evolve genomically over time. Some of the early models were thought to be these linear evolution models where you acquire a specific mutation, and then after that mutation is acquired, it has a very strong fitness advantage, and so it quickly overtakes the whole tumor mass. A more modern view is one of branching evolution, where we know now if we cut the tumor at any given point in time, you're likely to find multiple subpopulations. And the big question then is, are they competing in some way to drive tumor progression or maybe cooperating? There are also models like neutral evolution, which suggests that after the initial mutations, there's really just neutral drift in the tumor. And a model that I'll talk about a lot today called punctuated chromosome evolution, which suggests that most of the genomic evolution is occurring pretty early on. And after that point in time, clones emerge, and they're quite stable, and they just stably kind of expand to form the tumor mass. So there are really three different approaches people are using to study genome evolution and intratumor heterogeneity in the genomics world. The first one is to do deep sequencing. So you sequence a tumor very deeply, either by a whole exome or a whole genome. And then you cluster mutation frequencies, and you apply a lot of normalization for tumor purity, copy number correction, other things. And these are methods like SciClone, PyClone, many others. And they give kind of a broad, I think, general kind of overview of the number of populations that are present in the tumor. The nice thing is they can be applied to any existing data sets. The other approach is called multi-region sequencing. And so this was made very popular with Charlie Swanton's early work in kidney cancer. And here we're sampling from different spatial areas in the tumor and performing some type of genome sequencing method. And then the assumption is that the clones are spatially segregated. And so it works well in this example here when you see region 1 and 2. It doesn't work quite as well in region 3 and 4, where you can see the intermixture of different subclones. And then the third approach is single cell sequencing. And so this is where we've done a lot of work over the last 10 years. And here you can really, with single cell methods, completely resolve this intratumor heterogeneity and look at the genotypes of individual cells. And then you can compare those cells to understand heterogeneity and evolution. In the early days, we were challenged quite a bit by the limited number of cells that you could profile to do this with. But these days, those obstacles have largely been overcome with much more higher throughput methods where we can look at thousands, even tens of thousands of cells in a single tumor. So a while ago, when I was a graduate student, actually at Cold Spring Harbor, we developed one of the first single cell DNA sequencing approaches. And this used a combination of flow sorting nuclei. Nuclei are actually much easier to work with than cells. And we deposit those into 96-well plates. We use a chemistry called DOP-PCR, amplified the genome, and then calculated copy number from read depth across the genome. And this worked quite well. Originally, we did this on 454 sequencers and then on Illumina GA sequencers when they came out. And so there's no multiplexing. Every lane was one cell. And from this data, we could then reconstruct the genetic evolution of the tumors. Since then, we've developed a number of DNA sequencing technologies for single cells. Our initial work focused mainly on looking at copy number evolution. After that, we developed methods to do whole genome sequencing. These are using more MDA-based chemistries to do whole exome or whole genome sequencing. We then realized that's quite expensive, and you can only profile a limited number of cells. So now we kind of reduce the target space to about, more typically, we would look at around 2,000 cancer genes. It's about 5 megabases of the genome. More recently, we've developed tagmentation-based chemistries that can give us also single molecule resolution in single cells. And they work quite well. I'll talk a lot about this today. So what can you do with single cell DNA sequencing methods? You can profile cells, let's say mutations or copy number, and kind of cluster them and look at them in high dimensional space to understand how many subclones are present within a tumor. You can reconstruct lineages using phylogenetic methods and build kind of a chronology of mutations that occurred within the tumor over time. You can look at how clonal frequencies change over time, so in response to therapy or between the primary and the metastatic tumors, and understand questions about selection and drift. And you can also look at mutation co-occurrence and mutual exclusivity, which is actually very important in a clinical framework, because oftentimes when you're targeting tumors, you want to know which subpopulations actually have those mutations. In this example here, we have 2 different clones. One has PTEN, the other one has CHD1. And then clone 3 that kind of has all these mutations existing in the same population. But the tumor cells are not evolving on their own. They're surrounded by the microenvironment. And so the microenvironment is co-evolving with the tumor cells. And single cell RNA sequencing methods turn out to be a really great way to study the microenvironment as well. And so with these methods, we can understand, for example, in breast cancer, how we go from normal ductal and lobular structures to premalignant disease, where the middle of the ducts start to fill in with tumor cells, and you start to get some changes in the microenvironment, to kind of full-blown invasive disease, where the tumor cells are now spreading throughout the tissues, and you have a lot of changes to the immune and the stromal microenvironment. So single-cell RNA sequencing methods are really great tools for resolving this ecosystem of cells within the tumor, and you can typically measure tumor cells as well as immune cells like T cells, macrophages, B cells, and stromal cells like endothelial cells or fibroblasts. But in addition to just resolving cell types, which is quite easy to do, you can then take an individual cell type, like let's say T cells, you can recluster the data and you can get different cell states. And the cell states imply different biological functions. Some of them are known, like in T cells, a lot of these are well characterized, but in other cell types like fibroblasts or endothelial cells or even myeloid cells, those cell states are not that well established. You can also take the tumor cells and recluster the data and look at how signatures and phenotypes are varied across the populations. You can look at how different tumor cells might be in proliferative capacity, hypoxia, EMT, other phenotypes. So they're quite powerful methods. You can also look at expression subtypes and how they kind of coexist within tumors. You can do kind of pseudotime inferences on looking at developmental lineages in some tumors. And you can do cell type specific differential expression analysis so that you can only look at, say, the fibroblast before and after treatment, which is what is really powerful about these tools. So if we take a look at all the literature published so far on kind of cancer research studies, initially a lot of the work from our group, also from many others, focused on looking at clonal evolution in the primary tumor and also changes in the microenvironment. Following that work, there were more studies by Dan Haber's group and others, some of our work as well, looking at metastasis and comparing primary and metastatic tumors, which is still a pretty big area of research right now, as well as the circulating tumor cells, which are kind of the key intermediates in this process. More recently, work from our group and others are focusing more on premalignancies. In breast cancer, this is early stage DCIS, and then looking at therapeutic resistance, especially resistance to chemotherapy, immunotherapy, and targeted therapies, where we have to deal with other kind of technical problems like having limited core biopsy samples, but being able to get a wealth of information, I think, using single cell DNA and RNA assays. And then finally, being here at MD Anderson, we always like to think about how we can translate these tools into the clinic and for real important clinical applications to improve patient outcome and treatment. And so there's a number of areas that we've identified where we think these tools can really benefit. They range from early detection to risk stratification to discovering new drug targets in the microenvironment and the tumor cells, to also doing non-invasive monitoring to monitor in the blood, for example, how patients might respond to therapy. All right, so that was a little bit of an overview, and now I'm going to switch over and talk about one of our recent studies looking at triple negative breast cancer and understanding chromosome evolution during the expansion of the primary tumor. And this is work that we did with a graduate student of mine, Darlene Manussi, in the lab, Heng Yi, who's also a graduate student. We collaborated with Francisca McCord on some of the mathematical modeling that we did. She's at Harvard, and her postdoc, Michael Nicholson. And we also work with Peter Van Loo, who's at the Crick Institute, to do some estimates of genome doubling with our data. So the question that we were interested in is, how do you go from a normal diploid genome that has 2 copies of each chromosome to these complex aneuploid rearranged genomes that you see in breast cancer and other cancer types? How does this actually occur over time? And there are 2 models that we've considered in the past. One is a gradual model where you acquire chromosome event, let's say gain 1, then gain 2, then loss 3, then gain 4. And as you're kind of accumulating these in a stepwise process, the disease progresses and you go from, let's say, early stage cancer to invasive disease to metastatic disease. The other model, which we call punctuated chromosome evolution, is a model where you have this initial burst of genome instability, and so you get lots of rearrangements. And after that emerge many different genotypes, but one of those then kind of expands and forms a tumor mass. And so in this model, you're acquiring lots of genomic events at the same time. And after that, the rate of evolution is kind of geared down. The stepwise model really is the paradigm that you'll see in textbooks and quite abundant in literature. It really all started with Bert Fogelstein and Eric Fearon looking at colorectal cancer, where they described the stepwise series of events that occurred in colon cancer progression, where it started with an APC mutation, or also a 5q loss, DNA methylation, then KRAS mutations, p53 loss, and other alterations. And this was extended also very much to breast cancer and to cytogenetic data, suggesting that there really is this ordered series of events occurring over time as chromosomes evolved to generate these kind of full-blown aneuploid profiles that you observe at the end stage of tumor progression. But work by our group over the years has shown that, at least in ER-negative breast cancers, especially in triple-negative breast cancers, we see more evidence for this punctuated model where you have this initial burst of genomic instability, and then multiple clones emerge and they more or less stably expand to form the tumor mass. And the big question we had from this data is, although our initial kind of single-cell DNA sequencing technologies identify this punctuated model where early on you have this a lot of chromosome evolution occurring in a very short period of time, the question is what happens after that? Are the genomes really kind of completely stable as they expand to form the tumor mass, or is there still ongoing chromosome evolution but at a reduced rate? So to address this question, we wanted to develop some new single-cell DNA sequencing technologies. And we have this method called acoustic cell tagmentation. It's high throughput. It can be performed on 384 well plates. You can also potentially implement it on 1536 well plates. It's very short. It takes about 3 hours. And it's a very simple 3-step chemistry that uses transposases. And it's very cost efficient. So the way it works is we take nuclei, we extract them from frozen tumors or fresh tumors. We flow sort them into plates. And then we use this acoustic liquid transfer technology, which uses sound waves to transfer nanoliter droplets from one rate reaction into the other plate. And we perform a tagmentation chemistry. The acoustic liquid transfer system is really nice because it allows us to scale down reagents about 1 to 20-fold, which greatly reduces cost, and it automates everything robotically so that there's not too much kind of human pipetting occurring here. From there, we are able to perform this lysis of the nuclear membrane. We do DNA tagmentation, which means we fragment the genome, we add adapters on. There's a PCR amplification, and then we add dual barcodes so that every library has 2 unique series of addresses. We then pull together these plates and sequence them. So the chemistry is pretty straightforward. We're not the first to think about doing single-cell tagmentation chemistries. Actually, in a paper published by Andrew Addy, who's a faculty now at OHSU, almost 10 years ago, he showed that if you dilute DNA to concentrations that would be similar to single cells, I think he tried down to about 10 picograms, he could make sequencing libraries using tagmentation chemistry. Following that work, there was some nice work being done at Fluidigm, mostly by Richard Fakidi, showing that you could construct single cell libraries using tagmentation approaches. And then there was a bunch of work more recently by Saurabh Shah's group and Sam Aparicio, who developed methods like DLP, which use microfluidics for fluidic platforms to do these types of experiments. There was actually a platform around called 10x Genomics. They had a copy number based platform, and it wasn't based on tagmentation chemistry. It was based on multiple displacement amplification. But after about a year, they decided to kind of close down the product. So because apparently it wasn't making enough money, I think, for them, they wanted to focus more on other things. So that's kind of left a hole for a lot of people that started using the system. But I think the tagmentation-based approaches are really the way to go for anyone that's interested. So a big advantage of using tagmentation is that you can do what we call positional barcoding, which gives you single molecule information. When you do methods like DOP-PCR, which are the first generation single cell sequencing technologies, you're priming off of other strands. And so you can never know what the initial cut sites were in the genome where you perform the reactions. With the tagmentation-based approaches, you originally fragment the genome, and from there you're adding adapters and you're amplifying. So you know exactly where the cut sites are in that original, let's say, diploid genome, or it could be triploid or tetraploid for cancers. And then you do the amplification, and the amplification then will amplify the exact same fragment positions. So after that, you can remove all those types of duplicate molecules and reduce the amplification noise quite substantially. And you go back to the original, if it was a haploid or diploid genome, you would just have 2 copies of each sequencing read, or 1 copy. And so that really gives you very nice data to work with. We benchmarked this method against several other methods and found that it performed well in terms of the coverage breadth, as well as over dispersion, which is our metric of noise. This is just showing you one of the cells here from a triple negative breast cancer. You can see the data is distributed quite well around the copy number states here in this particular case. So we applied this approach to sequence about 1,000 cells from 8 triple negative breast cancers. Importantly, these were all untreated samples, so we're trying to look at the natural diversity here, except for the first one, which was treated with chemotherapy. And you'll also notice that the DNA ploidy is quite high for these tumors. They range from about triploid to tetraploid, which means that most of them did have a genome doubling event that occurred early on in evolution. So a few definitions here that I need to discuss in describing this data. We call these subclones that we identify as individual populations where a subclone is a single cell that shares an identical genotype or highly similar. And we also introduce this nomenclature of superclones. A superclone is in high dimensional space or in clustering space. These are groups of subclones that share a lot of common genomic features and are different than other superclones. And just to explain this a little bit further, we think our earlier kind of single cell DNA sequencing methods had the ability to resolve the superclones, but not really the subclones that we're seeing here within these populations. And then we also define different classes of copy number events. We have clonal CNAs, so those are ones that occur across all the tumor cells. We have subclonal CNAs, they occur in less than 90% of the subclones, but more than 2 subclones. And then we have what we call unique CNAs. Those are events that only occur in 1 subclone. And that's really, we have the resolution now to detect a lot of these more unique events. Here's just some examples of those, for different genes, which I'll discuss in a little bit more detail in the next slides. So this is the data showing you high dimensional UMAPs followed by clustering for 8 of these tumors, where we profiled about 1,000 single cells per tumor. You can see a lot of them have a number of subclones and superclones. Subclones ranging from about 4 to 25, superclones ranging from about 4 to 6 major populations. And this is showing you the distribution over here of the different classes of the CNA events that I just mentioned. So let me show you data for 2 tumors then. This is TN1. And so what you're looking at here is every single row is a single cell. We're looking at around 1,100 single cells. And everything that is in white is the baseline copy number. So that would be equivalent to the ploidy of the tumor. Anything in red is going to be more amplified, and in blue is going to be more lost relative to that. And these are the superclones shown here in the left-hand side, and these are the subclones here shown on the right-hand side. And so you can see there's quite a lot of genomic copy number diversity in these tumors, particularly between the superclone groups, but also within each individual subclone. And you can see that genes that we care about in breast cancer, like some genes like FGFR4, P53 are very clonal, and every subclone has the same copy number state. But other genes like cyclin B1, PIK3CA, about half the cells have 1 copy number state and half the cells have another copy number state. And then we see things like these unique CNAs where, let's take a look at EGFR over here. Most subclones are copy number 4, but we have a single population that has copy number 5. Here's MYC, for example. Most of them have copy number 4, but there's one population that has this copy number 3. And these are just similar results showing you this again for another tumor over here, TN2. And this is also what the other tumors look like. So next, in addition to the single cell sequencing, we performed exome sequencing, and we did this to be able to look at the mutations and to time whole genome doubling events. When we did the exome sequencing, we found that really the mutations are quite diverse across all the tumors, but there's one mutation, P53, which we see present in every single tumor, suggesting it's an early driver event in this breast cancer. That's consistent with the data from TCGA, where about 83% of triple negative breast cancers have P53. Next, from this data, we clustered the allele frequencies and we classified each mutation as being clonal or subclonal. And we then, you can see the kind of distributions over here. We then took the clonal events to try and estimate whole genome doubling through a collaboration with Peter Van Loo's group. And this uses basically LOH and timing in order to statistically estimate when the whole genome doubling event occurred relative from the diploid cell to the most recent common ancestor. In addition to that, we tried to estimate the timing of the p53 mutations, and we found that those always occurred prior to the whole genome doubling events in this data, suggesting that it may be necessary to have those mutations before you can start undergoing large chromosomal rearrangements. And you can see the whole genome doubling events, they roughly came in the middle of these timings and the lineages between the diploid cell and the most recent common ancestor. Now, this data was consistent with our previous data showing a large truncal distance here in the copy number evolution. But after the MRCA, what our data now shows is that there's additional ongoing copy number evolution occurring in most of these tumors. And so we call this the branching distance. And you can see it can be quite large. It can actually be almost equivalent to what we see with the truncal distance. We then focus in just on the copy number evolution occurring after the MRCA. And this is two different tumors here. From this, you can see that the copy number lineages are diverging. There's two major divergences here in this tumor, and then they split into the superclones and into the subclones. You're now looking at the copy number, which is not single cell data, but it's a consensus of the single cells representing the subclones. And from this, we can look at what the ancestral copy number profile looked like, which is the MRCA, which we estimate from this data. It's the minimal events that we observe across all these populations. And similar here, we see the MRCA split into two major lineages, and then further into the subclone lineage, superclone lineages, and then into the subclone lineages. And so basically what this data is showing you is that there's quite a lot of copy number evolution occurring after the MRCA, which suggests that there's an ongoing copy number evolution rate. So from this, we collaborated a little bit with Michael Nicholson and Francisco McCord because we wanted to explore two different possible models of the copy number evolution rates after the MRCA. So in these models, we have the punctuated events that occurred early on, and then we considered two different models, one where there was ongoing copy number evolution at kind of a flat rate, and another one, so we call this the gradual accumulation model, and one where there was a period of transient instability following those initial punctuated burst events. And so this is an elevated rate that then drops down and then reaches a basal rate. And so they run this, cell death, cell birth model to get millions of cells that would be equivalent to a tumor size. And they consider these two different mutation rates. And they run these simulations for several weeks. And then finally, they look at the frequency of the breakpoint distributions. And that's shown over here. And so basically what they're seeing from the two simulations are these two lines, one in blue, one in orange. And then our actual experimental data is over here in the black. For this particular tumor, which is TN5. And so what we found in most of the cases is that if you look at an AIC value of the gradual versus the transient instability model, most of the tumors, their chromosome breakpoint frequencies fit over here, kind of in this area. So suggesting that they support this idea of a transient instability model after the initial punctuated events. One tumor was kind of close, TN8, but it still fell a little bit more to the side of what we think is the transient model. So next we asked, what if we take a look at cell lines? Do they also have copy number substructure like we see in the triple negative breast cancers? And we looked at 4 different cell lines as shown here. These are very commonly ones used in the field to study triple negative breast cancer. And here's just showing you the data from one of those, MBA NB231. And indeed, we found that the number of superclones and the number of subclones was roughly similar to what we see in the human tumors. Which was interesting and somewhat unexpected for us because, of course, these have been cultured for a very long time. And next, we want to know if we take a single cell out of a culture and we expand it for about 20 cell divisions, that's the time it takes for one cell to fill a whole plate, can you generate an isogenic cell line from this? This is a common procedure that lots of people do in molecular biology. But what we found from this data is that even in this relatively short time frame of expanding to a full plate, we introduce a lot of new copy number alterations. And so we're not really able to make isogenic genotypes. Even during this short time, the cells intrinsically rediversify. So this, I think, puts into question a little bit these procedures and how well they can really establish isogenic populations. From this, we were also able to estimate the copy number evolution rate because they're about, we said exactly 20 cell divisions, and we could eliminate cell death here. And so we see about 1 CNA event per 4 cell divisions based on this data. Next, we want to look at how subclonal copy number correlates with gene expression changes. And so we did this by taking the parental cell line and expanding individual cells and then making these populations of cells. And now I just told you that the cells rediversified during that time, and that's absolutely true. But when you look at a bulk profile, it kind of averages everything out. And so the genotype that you still observe here corresponds pretty well to the initial ones in the parental population. So that's what you're seeing down here. These are the isogenic expansions in black, and then the single cell data is in the dots behind that. You can see we represented almost all the populations pretty well in this data. It's shown over here. These are now the bulk expansions. And then we also did RNA-seq, so we can then look at what's the impact of gene dosage from copy number to expression. Now, if you just take the RNA-seq data itself and you cluster it, it actually clusters pretty well into at least the 3 major subclone groups. And then we basically looked at how well then does that correlate kind of globally to copy number changes. And you can see a somewhat linear relationship where increasing copy number leads to increasing gene expression. And then next, we wanted to see the impact of specific subclonal events. And so this is showing you, for example, a region on chromosome 11. This is in the cell line MB231. And you can see most of the subclones had this big deletion here on 1q, but 2 subclones had kind of a retained chromosome here. And so when we look at genes like ATM and CBL, which are associated with breast cancer, you can see that they have increased gene expression relative to the other subclones. And you see that over here for CBL as well. If we take a genomic window of genes and look at the expression, you can also see that in the window, all the genes are kind of going up when you look at 100 gene intervals. This is another region on chromosome 19. Again, we see these subclones have a deletion over here on the P arm, and this blue one has amplification of PIK3R2, sorry. And if we look at gene expression, you can see the blue is going up here. The yellow is kind of down here. If we look at individual genes, you can see PIK3R2 is really kind of increased in expression here, whereas this other one, SH3GL1, shows a very low expression. So that's kind of within the subclonal areas, we're seeing this pretty big impact on gene expression. And then the question is, what about the rest of the transcriptome? Is it being impacted also in a cis way to these events? And indeed, we found that many of the cancer phenotypes that we typically study and are interested in, they do seem to be quite diverse across different expanded subclones that we generated in the cell line. So what we're seeing here then is we don't see evidence for a gradual copy number evolution model. We are seeing what we call a punctuated model. And then the question was, previously we thought that punctuated evolution, after those punctuated events, we thought that genome evolution was quite stable. And so there wasn't ongoing copy number events that were being acquired. And so we call this period clonal stasis. But what our new data suggests now, that when we have single molecule resolution, we can profile much larger numbers of cells, is that there does seem to be another period of transient instability occurring. And even after that, there's kind of a basal rate of copy number evolution. So we call this punctuated evolution and transient instability, and that's kind of illustrated here with this model. So to summarize this part of the talk then, we developed this tag mutation-based single cell DNA sequencing approach, which works very well and gives us also single molecule resolution. We find that in human triple negative breast cancers, there's a very large number of copy number subpopulations. We can now detect these subclones, whereas previously, I think we were mostly measuring the superclones. And early events that we see in evolution include P53 mutations, that's followed by genome doubling, and then you have this punctuated burst of instability. And then that's followed by this transient instability, and then an ongoing basal rate of copy number evolution. And interestingly, our data really suggests that at least in the cell lines we're looking at, there's really no such thing as isogenic expansion. So we think this has pretty big implications because a lot of people do isogenic subcloning, and then downstream they run functional assays, drug screens, lots of other things. And our data suggests that you really can't retain a completely isogenic genotype. You do tend to make the population more clonal, but again, even in those 20 cell divisions, they can kind of rediversify their copy number profiles, which then impacts gene expression. Okay, and so then for the last part of my talk, I'm going to switch over to a computational method that we developed pretty recently. This method is called copycat. It stands for copy number inference, copy number karyotype analysis of tumors. And this was a method that was really developed by a former postdoc of mine, Ruli Gao, who's now faculty over at Methodist Research Hospital. And we collaborated with a few different groups to apply these methods to look at single cell RNA data and the inferred copy number information with Stephen Lay, who works on anaplastic thyroid cancer, and Stacy Mulder, who works in triple negative breast cancer. So many of you probably work with single cell RNA sequencing data, especially from the 10x Genomics platform. We do as well. And a big question we have often is, when we take that data and we cluster it, we get different cell types, and some are immune, some are stromal, and then some are tumor, but there's also often normal epithelial cells present. And so we have typically a hard time identifying which clusters are really the tumor cells, if the tumor cells are present at all in low purity samples. This is just showing you a few examples where if you look at the epithelial markers, you can often find two different clusters. And you don't know which cluster are tumor cells and which clusters are normal epithelial cells, or you don't know if there's two different tumor subclones or two different normal epithelial populations. And this is common, aneuploidy is common in about 90% of solid tumors. So what we thought of here was, well, why don't we infer aneuploidy and look at the difference in diploid and aneuploid profiles, and then use that information to identify what the tumor cell populations are. There's previous work on this as well, actually very pioneering work that was done mostly by ATTIS, who was working with Aviv Rajev and also with Mario Suva. They showed a while ago in 2014 in this glioblastoma patient paper in Science that if you use moving windows of gene expression across the genome, you can identify some copy number variants, and you can use that to estimate especially whole chromosome arm level differences. And this is a really great method that a lot of people, I think, still use, and it's maintained very well at the Broad. But it also has some limitations. And so in using this method, we found that some of the limitations are that you need to have a matched normal sample that you're including each time with the other sample to try and eliminate some of the background noise that you're getting from gene expression. And it's really a moving window average. So it's hard to really identify specific chromosome breakpoints and segments, which we typically want to study when we're looking at copy number events. And then we also found that the hierarchical clustering doesn't work quite that well for identifying different subclones within the tumor. So really worked to develop this method, and it starts with a gene expression matrix that you would typically get from a 10x Genomics experiments. She then goes through some DLM normalization steps and also removes some outliers that we get that have very high kind of UMI counts. From there, what she does is she tries to cluster the data and identify which of these are likely to be the diploid cells. So most tumors are mixtures of aneuploid cells and diploid cells. And then from there, she uses this Gaussian mixture model to identify the diploid cells. And from there, we transform the gene expression, and then we apply a Bayesian approach that involves both a KS test to join segments so that we can determine which segments kind of belong together based on the distributions of those read count values. And she uses actually an MCMC-based approach to do this. After that, the segments are joined together into larger segments, and then you get whole segments across the whole genome. And then we look at the kind of transformed gene expression, and we can look at events that are neutral, losses, or gains across the genome. And then we cluster that data, and we identify populations that are diploid cells or aneuploid cells. And from there, she uses hierarchical clustering, and we can identify not just the copy number differences between aneuploid and diploid cells, like we show over here, but then also different subclones in the tumor. So this is showing two populations, one labeled as A, one labeled as B. And from there, we can then really look at the gene expression differences between those clusters. And this allows us to kind of link the genotypes and the phenotypes together from these single experiments. And the genomic resolution is roughly around 5 megabases. We tested out many different resolutions. If you go below that, the gene expression data is just a little bit too sparse, and you get a lot of technical noise coming from that. You could probably sequence at higher depths and things to overcome this and get lower resolutions, but for a typical 10x experiment, which is usually around 50K reads or 100K reads per cell, this works quite well. So we looked at around 20 different solid tumor samples. This includes anaplastic thyroid cancers, triple negative breast cancers, and pancreatic adenocarcinomas, as well as 2 ER-positive breast cancers. And we also looked at some of the older data sets from the glioblastoma tumors that were published in that previous study that I mentioned from ATTIS and others. We didn't spend a lot of time looking at liquid cancers. They don't have a lot of copy number alterations, so they can be quite difficult. And we also didn't really look too much at pediatric cancers, which is another cancer type that doesn't have a lot of copy number aberrations. In cancers like that, we don't expect these methods to work well. But in most solid tumors where there are copy number events, we do expect them to work. So this is then an experiment where we used bulk DNA sequencing data of a DCIS tumor to get kind of our reference, which is shown over here. We applied InferCNV, which is the moving window average approach that I just mentioned. You can see it over here. It does identify many of the events, but as you can see, it's kind of it doesn't give you exact breakpoints. It's kind of this wavy estimation of the density of the reads across the genome. And then this is copycat, and so you see here with the segmentation-based approaches, you get very kind of precise chromosome breakpoint information. And then over here, we estimated kind of the relative deviation to the bulk DNA sequencing approach, and we saw that InferCNV copycat did perform much better than InferCNV. And we also looked at gene interval stability. That's kind of like the intervals of the genes that we use. And we saw also that copycat performed better looking at those intervals. And so we applied this then to a few different cancer types. These are 5 pancreatic cancers. And this is up here showing you where we inferred the copy number as A, and that was aneuploid, and then diploid is in green. And so you can see here, there was a single aneuploid population that we identified with copycat. If you look just at the RNA data, you can see there were actually 2 to 3 different clusters here. And so this helped us say that this is actually a diploid epithelial population. This is the tumor population over here. Similarly, in the other ones, we often identify 2 or more epithelial populations based on a set of markers. And in our copycat data, we identified a single cluster as being aneuploid. So it helped us really identify the tumor cells here. This is the actual data from one of the experiments, one of the patients. And in PDAC, the tumor cell purity is actually quite low. And you can see that over here in these bars, but I think even in these samples where the purity was pretty low, we still identified these populations. And these are anaplastic thyroid cancers. Again, a bunch of cases where we applied copycat. And in some cases, of course, there was only one population that had epithelial markers. And so it was already clear that that was going to be the population of tumor cells. But in other cases, we had, for example, 2 clusters. And so this helped us identify which cluster is more likely to be the tumor cells. And here we see a lot of variation in purity. Some of them are quite high, but other ones are quite low. And so we worked with this group to try and really characterize these tumor populations. And then finally, we looked at triple negative breast cancers. And so in triple negative, we actually often detected multiple populations, both from the RNA expression data, but then from the aneuploidy inferences as well. And actually, interestingly, here, in fact, there were actually 3 different populations with epithelial markers. But there was 1 tumor population. But in some of these cases, we identified multiple tumor populations. And so these represent different subclones. And I'll show you that data in just the next slide here. But with the TNBCs, we see much higher tumor purity kind of across the board in terms of these samples that we analyze. So if we compare then our ability to identify the tumor populations just based on the copy number, and we compare that to the expression cluster that had the tumor markers, we determined that there was about 98% accuracy for identifying the tumor cells in these specific cancer types that we examined. There's a bit of variation. Sometimes it didn't perform quite as well. Sometimes it was more in the 90s. But overall, I think it did a pretty good job. And then next, we were able to use this data to really look at those different subclones within the tumor. And this is a triple negative breast cancer patient where you can see we identified 2 different subclones. This is comparing their copy number profiles over here by taking a consensus across all the single cells. And then we did a differential expression analysis between these 2 subpopulations. And we identified a number of pathways that were different between the populations. That was interesting, but probably the most interesting part here was that there was this high-level KRAS amplification occurring only in one subclone B. And if we look at the expression of KRAS, we also saw in that subclone that it was very highly elevated. KRAS amplification is actually quite uncommon in triple negative breast cancers, but it's interesting that we're seeing it here in a very small subpopulation. This was another triple negative where we also identified a KRAS amplification in a kind of rare population. This is showing you 2 major subclones. These are the consensus profiles. And then we did the DE analysis, identified a number of pathways that were different. But here you can see the KRAS amplification was only in clone B, and you can see increased expression here as well. So it's quite interesting. So to summarize, Copycat uses this Bayesian Gaussian mixture model and segmentation approach for copy number data from single cell RNA data, especially from high throughput methods like 10x genomics. Resolution is about 5 megabases, but again, that's dependent a bit on how deeply you sequence your cells. And we applied it to 3 different solid tumor types, and we're able to get pretty high accuracy in terms of identifying the populations just based on the copy number data. Of course, in reality, when you run these types of experiments, you probably want to look at both. You want to look at the copy number information, and you'd like to look at the gene expression. Then you get kind of a double confirmation that they're tumor cells. All this, the tool is available. There's a GitHub page, really maintains it very well. So people are always asking for feature requests and things, but anyone's free to use it, and always love to hear your feedback on using it for different cancer types. So in the last few minutes, I just want to mention some of the interesting future directions that we're thinking about a lot, and I think the field in general is thinking about. The first one is really in thinking about normal references for studying cancer. We do think it's very important to have these normal references when you're studying transcriptomes or epigenomes. And so we're quite involved in a project called the Human Breast Atlas, where we're identifying in normal breast tissues from women across different ethnicities, different backgrounds, different pregnancy statuses, et cetera, what are the normal cell types, and then what are their cell states? And that baseline is very important, both in the immune cells, the stromal cells, and the epithelial cells. And we're also contributing that data to the Human Cell Atlas project. And there's also the HubMap project, which is kind of the NIH version of this. And so these projects are really important. And I hope that people will really make use of this data for all the different organ systems when they're trying to study cancer. The next area is really multi-omic technologies, and this is an exciting area that there's a lot of work being done in right now. From our side, we're very interested in looking at DNA plus other things. So we're trying to develop technologies around doing both DNA and RNA sequencing in the same cell, DNA and epigenomics. And we think these methods are very powerful for linking together genotypes and phenotypes. So you can know how certain mutations or copy number changes are actually causing the cell to change in its phenotype, like in proliferation or invasion or other types of phenotypes. And there has been a lot of progress made in this area. There are some methods right now where you can do DNA and RNA sequencing or RNA and attack. They're generally low throughput though, and right now there's still quite a bit of technical error. So there's still a lot of development needed in these assays. 10x Genomics recently developed one for attack in RNA. The only problem is that the RNA is nuclear RNA and not cellular RNA, so it's a bit harder to work with. But this is a pretty exciting area, I think. And we're also very interested in spatial methods. So how can we link spatial information, especially from histopathology, and also morphology of cells to the genomic data, whether it's DNA, RNA, or epigenomics. And so there have been some new platforms developed like spatial transcriptomics, also SlideSeq. Those platforms, they work quite well and they give you pretty high resolution, but currently it's not single cell. So one spot on these arrays is going to be 50 to 100 cells. And so you still have this kind of mixture problem. There are also in situ technologies which give you single cell resolution, but then with those techniques, you're usually more limited to looking at a specific set of probes or targets. There are some in situ sequencing technologies now that are being developed, and some of them are pretty exciting. The only issue is that they have very small spatial resolution, so you can't look across centimeters of tissue or so. So still an active area of technology development that I think is really going to impact the field. And then finally, we're very interested in developing methods to look at archival materials in the clinic. Most clinical samples are FFPE materials. A lot of them are 10 or 20 years old, but the good news is that then you know the clinical outcome of that patient. Did they survive? How did they respond to therapy? Did they metastasize, et cetera? So a lot of work is being done by our group and others to really work with FFPE materials such that we can really repair DNA that might be damaged, and develop techniques that can analyze this tissue because it's quite a valuable resource. And the same goes for RNA, although RNA is even more difficult to work with. Even right now, there are a number of people working with single nucleus RNA data from frozen tissues, and that seems to work quite well. But the RNA profiles are just not comparable, I think, to what you get from looking at live cells. So still quite an area of active technology development. And so I think I'll end there and just want to highlight and thank all the people in my lab that contributed to this work. The first study I showed you involved work from Hanhui Yi and Darla Minusi, as well as Emise, Shan Shanpei, and others in the lab. The copycat method was developed by Ruli Gao, also with support from Alex Davis. TAPSI also contributed to this work. Our collaborators, Francisca McCord, and over at the Crick Institute, Peter Van Loo, as well as Toby Baker and Maxime, who contributed to the Nature study. And then all of our funding support, which of course helps us allows us to do this research. So thank you very much, and I think I'll stop there and take any questions.
Thank you, Nick. Excellent. That's fantastic talk. Thank you. So any questions? I think you can mute yourself. Feel free to unmute yourself to ask any questions you can have. Just get them shy. So, Nick, I have a question regarding the Nature paper. Actually, that was the paper selected as the journal club. Excellent. So, for the you have generated quite a number of variation profiles as well as the exome sequencing results. I wonder the concordance between the two when you build up the phylogenetic tree. The senior A phylogenetic tree is identical, or there's some subtle difference between the point mutation allele frequency build up trees?
Yeah, so there's no easy way to directly integrate those two data sets. We did look at this in some detail, but the problem is that the single cell DNA data is very sparse. You know, we sequence it at 0.1x depth or less. When you merge data together, if you merge single cells together across a lineage and then try to call mutations, it's a little bit better, but the problem is you become highly biased towards detecting the truncal mutations and not so much the small, more nuanced kind of lineage mutations. So it didn't work very well. We didn't try to infer, I think what maybe you're asking is, could we just use the exome data to infer a phylogeny, and how did that compare to the copy number phylogeny? Yeah. We didn't try that. I think it's still pretty difficult, at least the methods that I'm familiar with, inferring accurate phylogenies just from exome data alone. What we tried to do is integrate the two data sets, but the problem is that it was very biased towards truncal mutations. And we ended up focusing a lot more just on the copy number lineages, and then using the mutation data more to infer the timing of the whole genome doubling events, looking only at the clonal mutations, which happened before that MRCA.
Yeah, awesome. I think the transient instability model is really cool. But you actually tested this model just based on the 8 sequenced sample, right? I wonder if you checked the additional samples, if this model still holds.
Yeah, so we didn't look at a large number here. That's true. I think it would be interesting to look at it across many cancer types too, not just in, triple negative breast cancer. We think the punctuated model, although we don't know about the transient instability, might apply to lots of solid tumors, especially when you see p53 mutations and a genome doubling event that occurred that then brought that genome up to a triploid or tetraploid level. And so I think it'll be interesting to kind of apply these approaches across other cancer types, especially, solid tumors with p53 mutations, things like ovarian cancer, glioblastoma, maybe even colon cancer, a number of solid tumors. It's actually a bit of a different story, we think, in the ER positive breast cancers. They don't have p53 mutations, they have PIK3CA mutations, and the genome doubling is pretty uncommon there. So we think that might be a more gradual model. They don't have a lot of copy number events, 1P, 1Q, 16PQ, 8P. And so those might occur kind of more gradually over time. But when you see hundreds of copy number aberrations, I think in many cases, the result of those would have been that they accumulated in a kind of short period of genome evolution at an early stage of time. I mean, one of the big surprises, to us was that when we started to develop single cell DNA sequencing methods and sequence solid tumors, most, kind of the textbooks and literature said there's ongoing genome instability. And so every cell is going to be completely messed up, and different than the other one. And our data did not show that at all. We found that the single cells actually had near identical, aneuploid profiles that were quite complex. I mean, they had, hundreds of chromosome aberrations. but somehow those cells are able to maintain that genotype as they expand to form the tumor mass, going from a few cells up to millions of cells. And so that's what led us initially, I think, towards looking at this and coming up with this more punctuated model of chromosome evolution.
That's cool. And also, I think your cell papers also have that kind of flavor, challenging the traditional view of
I mean, looking at even early stage cancers like DCIS, we see that even there, all the genome evolution occurred early on within the ducts, even before the invasive tumor, started to expand from those ducts. So suggesting again that, not only copy number aberrations, but even most mutations are occurring early on in tumor evolution. So it's, I think it's quite interesting.
Hey, Nick, I have a question. Here's Jake. So very interesting talk. As you study the clonal diversity and punctuated model, I wonder if you can correlate the stable period with the tumor microenvironment or specific tumor type, or there's no such correlations and they're more like driven by the original mutations.
Yeah, it's we've thought about doing that type of work more with in vivo in vitro systems. The issue is that those punctuated events are occurring very early on. So when the tumor is either just a single cell or a few hundred cells or so, and so in a human tumor, it's going to be very hard to find those lesions. Even by the time a tumor is DCIS, it's expanded throughout the ducts, quite a bit. You can go back to early lesions like in breast cancer, it's called ADH, where you typically find a very, very small area with tumor cells, but most of those don't go on to form invasive tumors. So it's pretty difficult to look at, I think, the tumor cells early on in human tissues like that, and then the microenvironment, right? So if you're going to look at a lesion that small, if you want to look at the microenvironment, you'd want to look directly adjacent to those tumor cells. I don't think you would want to look at a whole, kind of tissue area. So it's difficult, I think, to track down those pretty early events. Later on, as the DCIS tumors expand, then I think it's much easier to look at the microenvironment and how's the immune cells impacted, fibroblasts and other cell types. But studying it really when that punctuated burst is occurring, that's going to be pretty difficult in human tissue samples.
And a technical question. So the ACT method, the resolution is roughly 1 megabase per, if I recall, right?
No, it's actually, we did that at 200 kilobases, but the method itself can go very, very high resolution. We've tested it down to even like 5,000 base pairs. And the coverage is very good because you get about 50% of the genome represented. So you can do very high resolutions if you want to. The trade-off is then you have to sequence much deeper coverage per cell. But so it might be good for some applications, like when you only have a few cells and you want really high depth data, like in prenatal genetic diagnosis, for example. But most of the time, I think people would prefer to have more cells. But yeah, the resolution can be completely tuned. It's not a limitation of the method at all.
Okay. How sensitive is that method? Have you validated or evaluated the sensitivity?
Yeah, in terms of detecting like a small population of cells. We haven't done like a more traditional like sensitivity test or anything for it. We're trying to implement it in higher throughput so that we can do thousands to tens of thousands of cells. At that time, I think it would be really nice to test, test sensitivity more and other things.
Okay, that's cool.
Hi, Nick. This is Chindo Higgs. Yeah, so I have a question about, so when you look at the diversity in the chrono-evolutionary process, how do you, what about if you look at different subtypes of triple negative cancer, is the evolutionary process uniform or there's also variability across the subtypes?
Yeah, that's a really good question. It'd be interesting, I think, to look across. So in triple negative breast cancer, there are roughly 4 to 5 different expression subtypes that people have identified. And, I haven't seen that many studies yet really looking at kind of, chromosome diversity or even chromosome aberrations that differ between those subtypes. It would be very interesting to look at. And I think in the current data, of course, it's just we don't have that many patients, and also we didn't run gene expression here, so it'd be hard to say anything about the current data that we have. But I agree. Yeah, it would be also interesting, I think, just in a very simple way, to look at the DNA ploidies and across the TNBC subtypes, because that would tell you about, are they all genome doubled or not? With triple negative, you see P53 mutations in about 83% of the patients. So what will be interesting to know is that if that other 17% or so, which tends to have more things like PIK3CA or other events, do they also undergo genome doubling? And is that associated with one subtype, like let's say LAR or something like that? So yeah, great question. We haven't really done a lot of work yet to correlate it with the gene expression subtypes, although we're very aware of those and we study those in a lot of other contexts.
Okay, that's great. So thank you, Nick, for the wonderful talk.